Hello everyone, good evening to all of you. This is going to be again another look at some of my notes that I took during the lecture in my class for this next set of chapters. They're about immunity, and I think the test is actually next week, so it's pretty fitting for the Halloween season that it's going to be pretty spooky unless we go and review a ton. So let's get started here with innate immunity, also known as the generalized immune response of the body against pathogens. So a little history. A scientist named Ilya Menchikov from Russia, aka the Soviet Union at the time. This was in the 1900s, so it's still going to be the Soviet Union. He found amoeba-like cells surrounding a splinter or a wound that he made into something. And the cells would ingest foreign material, so he called these phagocytes, or cells that eat. And from this discovery and these observations, he won the Nobel Prize in 1908. So this is going to put the foundations that there must be an immune system in our bodies that's defending us. Alright, again, there are two parts in the immune system. We're going to be focusing in this section on innate immunity, which is routine protection. And it's more generalized. It doesn't involve any pattern specifically. However, we do have recognition of specific molecules, even though it's non-specific, which is kind of a paradox in itself, but we're just going to put pattern recognition here. And then, of course, we have adaptive immunity, which will be covered in the next couple of videos. I also have some up there from class, but then I want to actually make my own as well. Okay, so adaptive. This is going to be much more specific, and this is going to develop throughout life. We're going to have antigens and antibodies. And then this is also going to involve the destruction of our own cells if need be to prevent spread of the infection. Alright, let's go into innate immunity with our first line defenses. Again, this is going to be about innate immunity, so that's just going to be routine protection. We're going to have these first line defenses of all exposed surfaces. That are lined with epithelium. That would include the borders being outside or inside the body, so outside would be the skin, and then inside, these first line defenses would be the digestive tract, as well as the respiratory tract. So these are going to be, again, our first line defenses. They all need epithelium. Okay, so in addition to that, we could also talk about the body's borders, which will be another physical barrier to all of these pathogens. Of course, our main one will be skin, and of course this is difficult for microbes to penetrate into. We have the dermis, which is going to be tightly woven connected tissue. Again, the skin, it's going to be defense in the sense that it doesn't let things burrow into it very easily. Then we also have the epidermis, which should be highlighting some of these terms here. Let's go and just highlight skin, because that's kind of the large category here. Epidermis should also be highlighted. We have a ton of things here to cover, such as the many layers of epithelial. And then most of the outside are dead. And they're filled with keratin. It's a protein. They're going to be repelling H2O, and this will create a dry environment. And a dry environment will be useful in preventing the growth of a lot of these moisture-dependent pathogens. Since the outside layer of the epidermis will be dead, they're going to slough off. And this is going to remove any attached microbes. And then the shedding again removes the microbes. This is actually pretty surprising that you lose 1 million cells a day, or 8 pounds of cells per year. Some very, very, very surprising statistics there. Alright, let's push on. The mucous membranes, another line of defense. If I recall correctly, mucous membrane is also the name of John Constantine's band. Like in DC Comics, John Constantine, I think he had a punk band named Mucous Membrane. It's been a while since I've read the comics. Anywho, so when we are talking about mucous membrane here in innate immunity, we're talking about the digestive, respiratory, as well as the genitourinary systems, or tracts actually. And there's going to be constant bathing in secretions, aka mucus. That's why it's a mucous membrane. You're going to be submerged constantly in mucus. The microbes are removed by peristalsis. That's in the intestine. And then we also have the mucociliary escalator. And that's going to be in the lungs, or the respiratory tract. We also have our own antimicrobial substances from our body directly. And these again are going to protect the skin as well as the mucous membranes. These will include salt from sweat. Not many microbes can grow in salty environments, so by having sweat and perspiration, we are kind of narrowing down what could grow on us and potentially danger us. We also have lysozyme. This is an enzyme that will be found in tears, as well as saliva. And this is going to degrade the peptidoglycan. Remember, peptidoglycan is seen in gram-positive bacteria. We also have another enzyme, peroxidase. And this is going to break down H2O2 into water and hydrogen, if I remember. Yep. I think that's what it is, or no, it was H2O2 is water and oxygen. Sorry about that. That's what it's going to do. It's just going to make it into those less harmful compounds. We also have lactoferrin. And this is going to bind iron. You might be wondering, why are we binding iron? That kind of sounds a little bit strange, right? So if we go up here, lactoferrin will bind iron, and this stops microbes from using this metal for their own nutrition and their own metabolic processes. We do need trace amounts of metals, and that's why if we're able to use lactoferrin and bind it and really hold it in there, then the microbes aren't able to use that nutrient. We also have something called AMPs. Anyone finds these, and you have to know all these different abbreviations when you're studying immunology. Well, AMPs are going to be your antimicrobial peptides. So these are going to be proteins such as defensins, which are going to make pores in the microbial membrane. We could just highlight AMPs there to remember it later. Alright, let's go into the normal microbiota, also known as the flora of the body. So we do have microbes just chilling on us at all times. Many of them are just harmless. They're just there, using your resources, being competitive, and keeping away actual harmful pathogens. But we do have this normal microbiota. It's kind of the reason why people take like probiotics and stuff. Alright, so again, they're going to be competitive towards pathogens. They're going to exclude them. They're going to cover binding sites and use up nutrients. So the bad bacteria and the bad microbes aren't able to use those nutrients. It just goes to the helpful or symbiotic bacteria. Another way they're going to be competitive and get rid of these bad actors will be the creation of toxic compounds. A species known as, or actually a genus known as Propionibacterium. These will actually degrade lipids to make fatty acids, which microbes do not like. E. coli will actually synthesize something called a colicin, and this is going to be in the intestinal tract. It's like a protein. I think it's just, again, a degrading protein towards other microbes, so it's able to compete. It's able to inhibit the growth of others. Then we also have lactobacillus. We've discussed lactobacillus before. This organism lives in the vagina. It lowers the pH, and lowering the pH, making it more acidic in that environment, will be from the metabolism of glycogen and from the mucosa. This will actually inhibit and sometimes even lyse other bacteria that can be harmful. Alright, we could also unfortunately disrupt the normal microbiota. How do we disrupt it? we are going to be misusing antibiotics, and this will, of course, predispose the person to infections from harmful bacteria such as Clostridium difficile in the intestine. And just highlight some of these. There we go. And Candida albicans in the vagina. And then, of course, the normal microbiota. They're essential to the development of the immune system. Alright, let's move on. Moving right ahead to the cells of the immune system. The main cells in the immune system, sorry about that notification or that advertisement, that was extremely rude, I apologize. Going to have blood cells, of course, and all the several types. Blood cells are going to originate from the hematopoietic stem cells, trying to spell it here, stem cells, and these are going to be in bone marrow, and when these are induced, they form colony stimulating factors, or CSFs. That's again one of our abbreviated terms there, CSFs, which are colony stimulating factors. Blood cells, of course, are going to move around the body through the circulatory. And we have categories. We're just going to list the names here. Erythrocytes. These are going to be carrying O2. We also have megakaryocytes. These are going to be clotting agents. And then we have our white blood cells, the leukocytes. Which are going to be important in defense. And then from the leukocytes, we actually have a ton of categories, which we'll be going over in a little bit. The first category in the leukocytes would be the granulocytes. And these are going to have cytoplasmic granules, hence their name. They're going to come from the common myeloid progenitor, and then from a myeloblast. So, from myeloblast. So they originate. And we're going to have three kinds of granulocytes. Again, granule, they have granules, cytoplasmic granules. The first kind would be neutrophils. And these are going to be the most abundant. I'm actually going to say that in a different way. There we go. And these are going to eat bacteria. The basophils are going to be allergic reactions and inflammation. They are going to be similar to mast cells, which are found in tissues. So this is going to be allergic and inflam response. And mast cells in tissues are going to be a similar kind to basophils. Right. Next, our last kind are eosinophils, and these are going to deal with worms. Parasitic worms are helminths, as well as allergies. So the basophils and eosinophils are going to also be involved in the allergic reaction. I need to just highlight that there. Our second kind of leukocytes are going to be the mononuclear phagocytes. This is another category where we are going to come from the monocyte. So let me see how I wrote that. And these are going to form something called the mononuclear phagocyte system. This is again one of our abbreviations, the MPS. They're going to include monocytes, which are going to circulate in blood, and then the cell types that they develop will be going through that process of development as they leave the bloodstream. All right, we have the macrophage, which we'll look into much more detail later on. They're going to differentiate. And then we also have the dendritic cells. These are going to be our scouts that engulf, and then they bring it to, they bring the material to the adaptive immune system. Remember, that's the one that's highly specific, that's built throughout life, and we have like antigens and antibodies. These are going to come from monocytes as well. Alrighty, our third group here, I guess we're not going to talk about them in this. Oh wait, there we are. Sorry, I got the slides mixed up. There we go. Our third group will be the lymphocytes. Again, lymph, that's like adaptive immunity. That's from the lymph nodes, all those lymphatic tissues. So we're going to talk about these ones more in the adaptive immunity videos. But these are going to come from lymphoblasts. And again, adaptive immunity. We're going to have B cells, as well as T cells, and natural killer cells as well. These are going to be specific recognition. Then more on them later. They are going to be found in, again, lymph nodes and tissues, lymphatic tissues. And then the natural killer, they're going to destroy certain cells. Again, more on them later. When we cover the adaptive immune system, we're going to go into such a large amount of depth with those ones. The T cells are going to be like helpers. They're known as, in the chart here, effector T cells. They're going to be extremely specific. The B cells will become plasma cells, and a lot of the accumulation of antibodies has to do with B cells and their interaction with the T cells. Again, more on that later. This is only the innate immunity, so we'll look at those in more depth in the adaptive immunity side of things. Alright, let's talk about cell communication. This is probably like the fourth time we've talked about cell communication at my university here, because I took cell bio and I also took biochem. We went through cell communication there, but there's all something different and all these interpretations of it. Alright, so communication, again, coordination. the big key here. Surface receptors will be like the eyes and ears. They're going to be spanning the membrane, and that's going to mean out to in. They're going to be the connecting mediators. We also have the adhesion molecules. These are going to allow cell to cell attachment. So blood, for example, blood vessel endothelial cells, they could actually make these adhesion molecules to bind to the phagocytic cells. And this will allow them to move to the tissues, or just in general exit the bloodstream. Alright, let's talk about some of the cytokines now. These are the voices of the cell. These are going to be made by cells to diffuse, but they get bind to receptors, and they have a ton of changes that they could create. They could cause growth, cell death, differentiation, and so much more. Movement as well. We could have local, regional, or systemic effect. A ton of types here, so let's go through them. Alright, a ton of types. Let's do it. We have chemokines. This is going to be the chemotaxis of immune cells. So that's going to be, again, we are talking about one of the effects is movement. Chemokines, it's going to be that chemotaxis of movement. We also have colony stimulating factors, which we've talked about already, CSFs. This is going to increase and differentiate leukocytes. That's going to be one of our abbreviations. I'm going to highlight that one. We also have interferons, or IFNs. These are going to control viral as well as inflammatory response. It's another abbreviation, so let's highlight it. Interleukins are going to be also known as ILs. These are going to be made by leukocytes, and these are going to be immunity in both innate and adaptive immunity. So they're going to be involved in both sides of this system. We also have the tumor necrosis on 1S factor, or the T and F. This is going to be inflammation response, apoptosis. This is going to, you need to act together with other components. for the correct response here. Okay, now let's check out the pattern recognition receptors. These are PRRs, and they're going to be on the cell surface, on the internal membranes as well, from things in the cytoplasm. They're going to be looking for three main substances. We're going to be looking for pathogen associated molecular patterns, or PAMPS. These include, actually, let's just highlight the whole thing, why don't we? This is going to be cell wall components, lipopolysaccharides, peptidoglycan, and lipoproteins too. Again, pathogen associated, we have lipopolysaccharide. That is going to be, of course, from gram-negative. Very strange to notate that gram-negative are going to be in that area. We're also going to have peptidoglycan that's going to be prominent in gram-positive. We could also be detecting microbe-associated molecular patterns. And then I'm going to just put quotation marks. It's the same thing. These are going to be known as MAMPS, though, MAMPS. These include flagellin, the protein that makes the flagella. We also have viral RNA. That's also going to be noticed by the pattern recognition receptors on the cell surface. Our final category of things that we could detect will be the danger-associated molecular patterns, or DAMPs. And these are going to be host damage indicators. So the DAMPs are going to be a cell saying, hey, we're in danger. However, microbes are going to be like the flagellin. Then pathogen associated, we're going to be looking at those things that are like from gram-negative, gram-positive specifically. Alrighty, let's now again talk about another type. We have toll-like receptors. These are TLRs, and these are going to be in the membranes of sentinel cells, also known as the phagosomes. So they're going to find PAMPs. Again, that's going to be like the cell wall components, lipopolysaccharides, etc. Next, they're going to then transmit a signal to the nucleus. Once that signal is sent to the nucleus, this is going to be creating gene expression. We could also have the inflammatory response or the antiviral response too. Right. Now we're going to look at PRRs that monitor the cytoplasm. And we have two main kinds here. I just want to draw that line because the toll-like receptors do not monitor the cytoplasm. However, these next ones, the NLRs and the RLRs, do. Our first kind would be the nucleotide binding oligomerization domain-like receptors, aka NOD-like receptors. So nucleotide binding oligomerization domain. That's all I'm going to write for that part there. NOD-like receptors. They're going to find bacterial components that mean a breach has happened. Red alert, you know? They're inside. Okay, RIG, also known as retinoic acid inducible gene cytoplasmic proteins. They're going to find viral RNA for an alert that again says breach. So one of them is going to find bacterial components. RIG is going to find viral RNA. So we're going to nod to bacteria, but we're going to feel that the viruses are rigging the cell, and there's going to be a breach from that. Again, these are going to be monitoring the cytoplasm, not the outside. All right, let's talk about the complement system. This is a very interesting thing. This is going to be where the adaptive kind of works with the innate immune system. I'm gonna make a smiley face there because they're going to kind of complement each other. There are proteins in blood as well as the bathing tissues that are going to be involved in this, and these are C1 through C9, and then C3 could actually split into C3A and C3B, so a whole slew of proteins. They're going to be activated by 3 paths. And then all these 3 paths will lead to C3 convertase, and then C3 splitting. Alright, so let's talk about those 3 pathways. We have pathway number 1. Let me see what I just wrote up here first before I start going all in this. I don't need to that much. Okay, let me find it. Or somewhere here. Maybe up here. Yeah. There, we have 3 different pathways. The alternative pathway, the lectin, and then the classical. So the alternative pathway is first. And in this one, this is going to be triggered when C3b binds to foreign surfaces. This is going to be very unreliable, actually, because C3 is so unstable that C3b actually already is there. Even when there isn't a foreign surface for it to attach to. The second one is the lectin pathway. And pattern recognition molecules, also known as the mannose-binding lectins. Or the MBL. They're going to bind to bacteria, mannose, and then cause a response. So they're going to look for the bacteria's mannose, their cells, and then a response will happen from there. Our third pathway will be the classical. And this is going to be activated by antibodies bound to antigen. So again, that's just extremely specific. There's a high amount of specificity whenever we're talking about antigen and antibodies. Okay, so once we use the pathways, so let's say we have alt, or the lectin, or classical. So all these will go to one place, which is C3 convertase. And then once we get to convertase, this is going to split C3 into C3A or C3B. Okay, so C3A, what happens with this form of C3 protein, this is going to be the inflammatory response. So A to inflammatory. The C3A and C5A will induce changes that contribute to local vesicular permeability and attract phagocytes. However, with C3B, we could actually have a few other things happen. We could have opsonization, which is basically marking the foreign particle or foreign object to kind of function as an opsonin, and this is going to make the phagocytic cells really excited. They're going to really want to eat it. So this is kind of like add sauce for phagocytes. C3B could also go down to a complex that's formed with C5, C5A, which actually C5A also goes up to the inflammatory response. But we also have C5B, let me write that in order, C5B, and we are going to get the lysis of foreign cells when we make this kind of a complex of all these proteins of the and then of these other proteins, and they find the membrane, they're going to start like poking it. So those are the potential outcomes of C3A, again, to the inflammatory response, to attract phagocytes. C3B will lead to opsonization, which is again adding the opsonin to the foreign cell, and then the phagocytic cells are going to get really excited and go after it even more. And then C3B could also join up with the other C proteins and lyse the foreigner by poking and penetrating it. We're going to be making something here called the membrane attack complex, or MAC. This is going to be assembling the complex in the cell membranes of gram-negative bacteria. The inflammatory response will actually also release cytokines. From the mast cells. And opsonization, we kind of already covered it one very well. Okay, let's talk about phagocytosis and the different stages in phagocytosis. This is again the engulfing action. So again, phagocytes engulf and digest materials and pathogens. There are several steps to this. We have first chemotaxis, and the recruitment is by chemo attractants. Okay, step 1. Step 2 would be recognition and attachment. This is where the direct attachment, which is going to be, you write these out, this is going to be using mannose binding to the receptors, or indirect, which is going to be binding to opsonins. Remember, the opsonins, those are the proteins on the outside, like a coating. It's like a sauce that allows for the phagocytic cells to become more excited and they go after it more. So indirect would be that process. However, direct would be just binding to those mannoses on the bacteria. The receptors are going to bind to those. Right, number 3, we're going to have engulfment. This is basically what it sounds like. The pseudopods are going to be working here. And then when they surround, you're going to form the phagosome. Next, we have maturation and phagolysosome. In this step, this is where we're going to get the endosomes fusing and the pH is going to drop. So basically the phagocytic cells are going to kill things by burning them alive in acid. The lysozymes are also going to bring more enzymes to help break things down even further. So again, they just burn the bacteria, cells, or pathogens in acid, and that's how they get rid of them. You also have step 5. This is destruction. We didn't even need more destruction, but we need to make sure we get rid of those pathogens. We're going to have toxic, reactive oxygen species enter the play, as well as nitric oxide. The enzymes are going to be there. Defenses are also going to be there. Defensins, again, make pores in the membrane. So pores, and then our lactoferrin will tie up iron. So it's actually not going to get rid of it, but I'm going to put like a lasso around it. It's going to tie it up. Hey, partner, buckaroo, where are you going? That's the step that we're going to be using our lactoferrin and tying up all those nutrients. And then the final step, number 6, is exocytosis, which is again, the vesicle will fuse with the membrane and out go the remains. So just spits out whatever it's finished with. Okay. Macrophages. Let's go back to macrophages again. We were up here with macrophages earlier, but we didn't go into that huge depth with them. These are going to be scavengers and sentinels. I'm going to actually just copy this over and move it down to where we left off. Less work, look at that. There we go. Macrophage. Mononuclear leukocyte again. They're going to destroy invaders, of course. and eat up dead cells and debris. They're going to be always in the tissues, and they call reinforcements. So they have these toll-like receptors. We talked about those on the surface. Again, I'm going to highlight that maybe because we did revisit toll-like receptors. Those are going to be on the outside. They're not in the extracellular matrix of the cell itself in the cytoplasm. They're just going to be on the outside. Let's go and find them. They're pattern recognition receptors. Yes, there they are. They're not in the cytoplasm. They're monitoring the outside. They're finding those PAMPs. Remember the pathogen-associated molecular patterns like the LPS or the peptidoglycan. Okay, so they're going to be also in the phagosomes. And these are going to ID invaders. Like we said, they're going to be looking for those PAMPs. The cytokines, the voices of the cell again, are going to be produced as the response. Okay, so an activated macrophage, this is caused to increase power. If they fail, let's highlight that. Activated macrophage is a vocab term. They fuse to form giant cells, or they could also work with T cells to form granulomas. Granulomas are going to be kind of like a barrier. They're going to wall off the organisms. And an example would be tuberculosis. It doesn't get rid of them, it just walls them off, keeps them in one spot. Alright, let's now talk in depth about the neutrophils. These are going to be a granulocyte of the leukocytes. Yeah. Here we said they're going to be the highest number, and they eat bacteria. So let's go into depth more about these cells. So this is going to be a rapid response. They're kind of like a strike team. They're going to move from the bloodstream to tissues extremely rapidly. They're going to be in the early stages of inflammation. They're more powerful than the macrophages. Again, they're like a SWAT team. Alright, they're going to be releasing the granules as mode to attack. They could also make these neutrophil extracellular traps or nets. And these are actually going to be made out of DNA, which sounds amazing. And then this allows usage of enzymes to destroy the invading pathogens. Alright, now let's actually talk about inflammation. know it's been such a long video. Inflammation, the inflammatory response, the purpose would be containing damage. Inflammation, you're going to contain the damage. Restore tissue function. And the biggest thing, it's local. It's gonna localize that damage. Results in swelling, pain, and heat. And sometimes loss of function. The pattern recognition receptors that will be important in this are going to be the TLRs and the NLRs, which are nod-like receptors. So again, this is going to be cytoplasm. This is going to be outside. We were talking about that. They're going to find the pathogen-associated molecular patterns, so PAMPs. Again, a ton of these abbreviations, and DAMPs. Again, DAMPs are going to be host has problem. PAMPs again are going to be like the lipopolysaccharides or the peptidoglycan. Again, a lot of these abbreviations. Look at this: the particle recognition or pattern recognition receptors, specifically in the inflammatory response, will be toll-like, as well as the nod-like receptors. And they're going to be looking for PAMPs and DAMPs. These are going to release the host will release the inflammatory mediators. These are going to be our cytokines, histamine, and the TNF. Again, that's the tumor necrosis factor. That again ties into the host having a problem. Inducers include tissue and microbe damage. So, damaged tissue. And this is going to cause a cascade of events. Cascade would be including a dilation. I'm going to actually just move down here. So dilation of vessels, the blood vessels. When there's increased flow, this is going to be heat and redness. That's going to be slower in capillaries. And then leakage of these fluids will cause the swelling. These are kind of like the reasons why we have those symptoms, the results. The leukocytes will go from blood to the tissue. This is where the endothelial grab. They grab the phagocytes, slow them down, and then this is going to cause a motion called diapetesis. This is when the leukocytes, or the phagocytes, the mononuclear phagocytes, are going to go and squeeze to tissues. Diapetesis is that the process. clotting factors, wall off the site of infections, and then the dead neutrophils will accumulate as the pus. Okay, let's talk about acute versus chronic. Acute inflammation, this is going to be short term, mainly neutrophils and macrophages cleanup. In chronic inflammation, if acute fails, this is when we get to the macrophages being the main player. The giant cells also form. Remember, when the pathogen can't be removed, we're just going to make more powerful macrophages. We're going to make the giant cells, and then the T cells, of course, are going to make the granules. And then once we get to those granulomas, we're going to wall off the invader. Granuloma, that was another vocab term we've seen recently. Alright, fever. Now we are going to talk about fever. This is the strong indicator. It's not local, it's everywhere. It's for infectious disease, especially bacterial. We have a temperature regulation center in the brain, and this is usually at 37 degrees Celsius. However, we could get this increased because of pyrogens. Pyrogens come in two categories. We have endogenous. Those are cytokines that are made by macrophages. And then after they detect the microbial products. However, we also have exogenous, where these are going to be made by the microbes themselves, such as the lipopolysaccharides of gram-negative bacteria in their outer membrane. So endogenous is from itself, the cytokines are your own, from your own macrophages. Exogenous, those are from the foreign pathogen. Fevers are also not localized. This is going to be wide spread, not in just one area. We're going to experience chills, not only because it's Halloween, but because the body temp is set below, and then we try to warm up. The effects of the fever would be the decrease in growth rate of bacteria, the temperature increase will also enhance all these different responses. We have inflammatory response, phagocytic activity, lymphocytes. This is all again because of the heat. We're going to increase lymphocytes, release neutrophil attractors. We're going to make interferons. We're also going to make antibodies, interferons, if we go all the way back here. they're going to control viral inflammatory response. They're a type of cytokine. And we're going to also release leukocytes from the marrow. So all those are effects of the fever, which is, again, widespread. It's not localized like inflammation. All right, that was innate immunity. Thank you so much for tuning in. Please have a wonderful, happy Halloween, and please do something nice for someone. Good night, everyone. Thank you so much.